Hey guys, today I'm going to be demonstrating how to make, place, and conduct transect plant sampling in order to identify plants using materials you probably already have at home. And at the end of the video, we'll go over how to interpret your findings. The materials we will be using for today's experiment are yarn or twine, a measuring tape, a ruler or meter stick, a set of sticks of some kind, could be popsicle sticks, kebab, chopsticks, or even sticks you find outside. So long as you can tie your transect string on one end and push the other end into the ground, it'll work. You'll need field guides for plant identification. You can find some online and print them out to bring with you. Lastly, you'll need your field journal to record your findings, and you can either draw or print out this chart. Pause the video here if you'd like to draw this table in your journal. You should have plenty of room on just one page. I also added an extra column on the left so that when I sketch a plant in my field journal, it is easier to reference which one of my data I'm drawing, since I might not know the name until later. Now we are going to make our transects. If you have a measuring tape, make your transects 10 meters. If you don't have a way to accurately measure meters, I'll show you how to use a ruler to make 33 feet transects. 10 meters is approximately 32.8 feet, so we'll round up. In research experiments, exact measurements are significant, and the metric system is what would be used since it is universal. To begin, take your yarn and draw a small line with your marker a few inches from the end of the rope. This will act as our actual starting point so that we don't lose any length when we tie it to our sticks. Begin wrapping the yarn around the ruler. Place the mark you've just made at one end and wrap the string around the ruler 33 times. Before cutting your string, make another mark at the end so you know where to tie your stake. Now simply tie your yarn onto your stake and line up the knot with the dot you drew earlier. It may take some adjusting, but try to get the dot as close as you can to being where your knot is, so that your measurements are as close to exact as possible. Now that you have your transects ready, it's time to find somewhere to place them. Since we're doing field work, wear closed-toed shoes and long pants to avoid bug bites or scratches from plants. Also, ensure you're not placing your transect on or near any poisonous plants. Look for areas that have diverse plant life, and if you have multiple groups, place them in different areas from each other. Try to consider the different factors that can affect plant life, such as landscaping, foot traffic, altitude, or distance from water. Once you've found an appropriate location, place one end into the ground and make sure the rope is pulled somewhat taut before placing the second stake. You don't want to have too much slack in the rope because that shortens the distance and can affect your results. As you consider the plants along your transect, think back to our observation chart and try to find somewhere that has plants on the ground, middle, and upper sections. You will be recording everything within a 5 centimeter or approximately 2-inch distance from the twine. Use a ruler to get a general idea of that distance. Let's take a look at this plant on ground level. Remember, we're only observing living plants. Our data should reflect current conditions. If I can ID it using my field guide, I'll write the scientific and common name. The description I would write would include the length of the leaves, the texture, what the margin of the leaf looks like, and I'd give it a name I can reference later, something like small vine with leaves of 3. If your teacher allows, you can take a small sample to identify later. In your field journal, draw the plants you observed on your transect, making note of things like the leaf shape, the texture or pattern of the leaf edge, and the approximate size of the leaves or other parts of the plant. Once you've made an entry for a plant, every time you see it, make a tally in the individuals column. This tells us the number of individuals, or abundance, of each species along our transect. Some field guides are organized by the color of a plant's flower, like this one. These are as simple as trying to find an image that matches what you see. However, if you are conducting this experiment when flowers aren't blooming, this kind won't be of much use to you. You may also use dichotomous keys, which involve answering a series of questions about what you see. Some are more specific than others, requiring you to know the parts of a plant and how to identify them. Suppose we see this tree on our transect. Let's try and identify it using this dichotomous key. Question 1 asks if the plant grows on trees or grows differently. Our plant is a tree, so it grows differently. On to question 3. Leaves are small and clumped, resembling a green rat tail, or different. Different, so on to question 4. Plant is spiny with pencil or pancake stems, like a cactus, or different. Different. On to question 6. Leaf veins run parallel or branch from a midrib. Let's take a closer look. They appear to branch from a midrib, but suppose you couldn't tell. The perks of using a dichotomous key is that if you find you arrive at an answer that doesn't seem right, you can always go back to the point you weren't certain and choose the other option. From 6b, we go to question 10. Are the leaves compound or simple? Let's look at another image. This is a preserved branch with leaves from our mystery tree. Compound leaves have many small leaflets on one stem that then attaches to the main branch of the plant. Simple leaves attach directly to the main branch, like in this image, which is a preserved cedar elm tree branch. You can see here that each leaf is attached to the main branch, but in our example, they are small leaflets attached to a single stem that is separate from the thicker, more woody tree branch. Choosing compound leaf sends us to question 11. Leaves are trifoliate, having 3 leaflets, or not. Ours has more, so we go to question 13. Leaves are once compound or twice compound. While it's easy to tell from the images which answer is correct, it's important to properly identify it using the information given, since some keys may not have pictures, or the species may look visually similar. If we look back at our earlier image, we can see that there are 2 leaves per stem, so it's twice compound. So our mystery tree is the honey mesquite. As you are making your observations, it is also useful to note any insect or animal life you find on your plants, as that can also help you identify them, since certain insects often like to live on certain plants. After you've walked the whole length of your transect and recorded your observation, it's time to interpret your findings. Using this chart, we recorded the number of different plant species we observed and how many there were of each kind. This concept is known as biodiversity. Biodiversity is a good indicator of the health of an ecosystem. Total species is represented by the number of separate columns you've filled in, and the percent abundance of a plant measures how common or rare it is compared to all other plants in its environment. Choose one species and calculate its percent abundance by dividing the total number of individuals of the one plant by the total of all plants and multiply it by 100%. One important fact to remember about scientific studies is that there are always limitations, which are parts of an experiment that could not be controlled that may affect the reliability of the results. Think about your study. What were some limitations you encountered? For me, one major limitation was that I had to conduct this study by myself, so there wasn't anyone to help me ID or look out for any potential mistakes I may have made. One could be your location. How does the location you chose influence biodiversity? Try to think of things that were both in your control and things that maybe you couldn't control. I hope you learned a lot from today's lesson. Thanks for joining me.